Okay, this is lecture 8. This is lecture 8B, part 2 on renal physiology. Earlier, we were discussing intrinsic control mechanisms for control of GFR, but there are also intrinsic to the kidney. These are also called autoregulatory. But there are also extrinsic controls which can regulate the glomerular filtration rate, or the GFR. And in that way maintain a steady GFR. There are two types of controls. One is mechanisms, extrinsic controls. One is through the sympathetic nervous system, or the sympathetic nervous, the sympathetic division of the autonomic nervous system, and the other is through the renin-angiotensin aldosterone mechanism. Now, in terms of sympathetic control, when the autonomic nervous system sympathetic output is at rest, that means there's no upregulation of sympathetic output. The renal blood vessels are normally maximally dilated, and autoregulatory mechanisms are the dominant regulatory mechanism that maintains a steady GFR. However, if the sympathetic nervous system is activated because of some stress, and by that I mean some trigger for upregulation, and there's many things that upregulate sympathetic nervous system output, then a number of things happen. One thing that can activate the sympathetic nervous system is a drop in the systemic blood pressure. So if systemic blood pressure drops, that's detected by the aortic bodies and carotid bodies, and the signal sent up into the brainstem, and that activates the autonomic nervous system to increase sympathetic output. The result is that norepinephrine will be released by sympathetic fibers that innervate organs like the kidney, and also that epinephrine can be released from the adrenal medulla. The adrenal glands in the center is the medulla, and this is an area that produces adrenergic type of neurotransmitters that go into the bloodstream. So as a result, vascular smooth muscle will constrict and blood pressure will rise. So in general, if your blood pressure drops too low, you have this kind of sympathetic output, and there's overall vasoconstriction, and the systemic blood pressure rises appropriate in response. Now, the afferent arterioles in the kidney will also constrict in response to that, and the glomerular filtration rate will decrease. And that lower glomerular filtration rate will help to raise, in general, the blood volume, because less filtrate will be formed, and therefore it helps to maintain blood pressure and prevent losses of fluid that could reduce it. So that's a direct simultaneous effect of norepinephrine being released from the ends of axons of sympathetic fibers, and epinephrine released from the adrenal medulla, causing overall systemic vascular smooth muscle to constrict, raising blood pressure, but also causing afferent arterioles to undergo vasoconstriction, thus decreasing the net filtration pressure in the glomeruli, and therefore the glomerular filtration rate. Okay, let's move ahead with this. The other extrinsic control mechanism which can have an effect is the renin-angiotensin aldosterone pathway. It's the major mechanism for the body to increase systemic blood pressure. If the blood pressure drops too low, then the glomerular filtration rate it drops. Filtration, in fact, will slow down inappropriately below the normal steady levels. This may be due to blood loss or dehydration. Severe blood loss, severe dehydration can cause that. The end result is a triggering of release of renin from juxtaglomerular cells. Renin, in fact, can be released by any one of three pathways or three stimuli. Reduced stretch of juxtaglomerular cells by a drop in the mean arterial pressure of the afferent arteriole. Stimulation of juxtaglomerular cells by the macula densis cells in the distal convoluted tubule, which would be activated if they detect or respond to low sodium chloride levels in the distal convoluted tubule fluid, which would be the case if you have a low GFR. Low blood pressure leads to low GFR, leads to fluid being in the tubular system for too long and too much sodium being reabsorbed from it. Therefore, by the time it gets to the distal convoluted tubule, the sodium levels are too low, and that will be detected by the macula densis cells. They'll stimulate juxtaglomerular cells to release renin. And the third way is by direct stimulation of receptors on the juxtaglomerular cells which respond to adrenergic signals. Adrenergic neuroreceptors will bind to beta-1 adrenergic receptors on the juxtaglomerular cells. And those neurotransmitters are released by sympathetic nerve signals sent along efferent sympathetic nerve pathways coming from the central nervous system. I think on the next few slides I go into each one of these three. I've already said it, but we'll review it further in detail. Let's see. Oh, no, I go ahead with it. Okay. Excuse me. So, the renin-angiotensin mechanism is a major extrinsic control triggered when juxtaglomerular cells release renin. Renin, as I told you, is a hormone protein that can act as an enzyme on a protein in the blood plasma called angiotensinogen. Angiotensinogen, the action of, and this protein ends in OGEN, indicating by its name that is in its inactive form. So renin acts on angiotensinogen, cleaves it to form angiotensin 1. Then angiotensin 1 is converted to angiotensin 2. It's converted over to angiotensin 2 by ACE, or angiotensin converting enzyme. I believe that this is ACE1. Just as an aside, ACE2, I believe an isoform of angiotensin converting enzyme 1. ACE2 is actually the target molecule for binding of COVID-19 to cells that express it. And ACE2 is expressed as a protein on the surface of some cells. It's also an angiotensin converting enzyme, but it can be used by, viruses use different types of molecules on different cells to be able to bind to them and then penetrate. And angiotensin 2, unfortunately, is not only found on the epithelial cells in the lung, but also in kidney cells and cells in the gastrointestinal tract and the liver, suggesting that the virus may also infect those and have an effect on those cells, not just lung epithelial cells. Anyhow, let's get back to this, which is angiotensin 2 converting enzyme 1, or ACE. Angiotensin 1 is converted to angiotensin 2, again by having enzymatic activity, which can act on, again, because ACE has enzymatic activity that can cleave angiotensin 1 to form it into angiotensin 2. Now, angiotensin 2, and I've talked about this before, it has a ability to trigger quite strong systemic vasoconstriction, thus raising overall blood pressure, causing mean arterial pressure to rise. It also has an effect on the adrenal cortex to stimulate the release of aldosterone. And aldosterone, as we've discussed before, will increase sodium and therefore also water reabsorption. It's resorption or reabsorption from the fluid in the tubular system. As a result of this, both systemic and glomerular hydrostatic pressure rises. Let me just think about this. Yeah. As a result of these effects, the systemic vasoconstriction and release of aldosterone causing more uptake, more reabsorption of sodium and water. You know, if you take up more fluid and salt, you'll increase the volume of the intravascular space and therefore increase the pressure. So both of these can return systemic and glomerular hydrostatic pressure back to within the normal range. So this is a diagram which summarizes a lot of what I've been saying, and well, let's go through it. Okay, so regulation of GFR, and here we're seeing that remember all of these pathways, these homeostatic regulatory mechanisms, the pathways always kick into gear when stimulated by some change in a key variable. And the variable here we're talking about is blood pressure, and the change is that blood pressure goes to an abnormally lower level, all right? And therefore mechanisms kick in to regulate it, because if the blood pressure is too low, then the GFR will be, if the blood pressure is too low, then the net filtration pressure in the glomerulus can drop too low, and therefore the glomerular filtration rate can be too low. So the mechanisms include intrinsic and extrinsic mechanisms, as we just spoke about. The intrinsic mechanisms are autoregulatory and include the myogenic response and the tubular-glomerular mechanism, feedback mechanism. They also include the other type of mechanism are extrinsic to the kidney, extrinsic mechanisms, which include the renin-angiotensin aldosterone pathway or mechanism, and neural controls, external neural controls involving the sympathetic nervous system. In terms of intrinsic mechanisms, you have myogenic autoregulatory responses of the vascular smooth muscle in the kidney, and vasodilation. If in the case of a low blood pressure, you can get vasodilation of the afferent arterioles, leading to increased blood flow into the glomeruli, causing a rise in net filtration pressure and therefore an increase in the glomerular filtration rate back into the normal range. At the same time, there is also this tubular glomerular mechanism of autoregulation, where you get, in the case of reduced filtrate flow, and now we're talking about filtrate flow in the distal convoluted tubule, either reduced flow or reduced osmolarity, because, well, going hand in hand with that reduced flow and reduced GFR, triggering the macula densa cells of the juxtaglomerular kidney to release vasoactive compounds that can also cause vasodilation, just as here you get vasodilation. So another mechanism to cause vasodilation of the afferent arterioles, thus raising, I think I'll change the color, thus raising, I didn't change it enough, thus changing here, let's make, I don't like that color, let's make it dark green, that's blue, thus vasodilation of the afferent arterioles, thus raising the reducing resistance, raising flow through the afferent arteriole into the glomeruli, through the afferent arterioles into the glomeruli, thus raising net filtration pressure and therefore raising GFR into the normal range. Now let's go over to extrinsic mechanisms. Normally intrinsic mechanisms are adequate for control and regulation of GFR, but if they're not, then the renin-angiotensin mechanism is one of the mechanisms that can kick in and and act. And in this case, the juxtaglomerular cells of the juxtaglomerular apparatus will be stimulated to release renin, and renin will act on plasma protein called angiotensinogen to convert it into angiotensin 1, which, and it's not shown here, but it goes to angiotensin 1, which then gets converted to angiotensin 2 by ACE in the lungs. And then angiotensin 2 has an effect on the systemic overall circulatory system, the systemic vasculature, to cause vasoconstriction, thus raising resistance and therefore raising systemic blood pressure. And when you raise systemic blood pressure enough, you will increase the net filtration pressure going into the kidney and therefore increase the glomerular filtration rate. The other thing that angiotensin 2 does, among other things, is it will cause, let's do that in another color, by the way. This, let's put this in another color, a third one. It can also trigger aldosterone release from the adrenal medulla, where that hormone is produced. And the aldosterone will have an effect to increase sodium reabsorption in the tubular system of the kidneys, therefore also taking water. Water will follow where the sodium goes, pulling back water, thus increasing the blood volume and therefore the systemic blood pressure, and thus a fourth way here, I'll put a fourth color, a fourth way to increase glomerular filtration rate and bring it back into the normal range. Neural controls are also part of that extrinsic mechanism to maintain proper GFR. With lower blood pressure, even if there's no problem, even if the the initial stimulus is low blood pressure detected by baroreceptors in the blood vessels of the systemic circulation, and you know that those baroreceptors, pressure receptors, are found in the aortic body and the carotid bodies, and that sensory info gets sent into the central nervous system, gets processed, and you get increased outflow of sympathetic division of the autonomic nervous system, triggering overall systemic arterioles to vasoconstrict, causing an increase in resistance, therefore an increase in systemic blood pressure, and therefore an increase in net filtration pressure in the glomerulus, and thus an increase in the glomerular filtration rate. There is one thing here I didn't mention, which is a crossover here. Macula densa cells, stimulation of macula densa cells also has a crossover connection to the extrinsic pathway, and that can trigger juxtaglomerular cells to release renin, okay, which is shown here in this part here. So that's a pretty comprehensive slide. It actually doesn't show everything. I'm sure some of you, or all of you, I don't know, are bowled over by the complexity of things that can occur in this kind of regulatory mechanism. Partly it's because of the key importance to normal physiological function of the renal activity or glomerular filtration rate to maintain proper water balance, proper electrolyte balance, and proper overall blood pressure by putting out more or less water, putting out more or less sodium, maintaining things in a homeostatic balance. So this is, quite an intricate way that it's controlled, and more than one control mechanism is operating. I can tell you that these are fundamental, basic. This is a fundamental, basic overview of the renal regulation. It is, in fact, even more complicated, and there are more subtleties to it, many more. And in fact, I can say with quite a bit of confidence that I'm leaving out a great deal, and many things are not completely understood, actually. I have it on good authority, more than one good authority, that it's actually way, way more complex than this. But this gives you a good fundamental idea of the background mechanisms that are occurring. Okay, let's shift a little and talk about other things. I don't know, I can do this within this part 2. There are, some things that won't get reabsorbed. Sometimes, often that's good and that's normal. Sometimes it's not good. And it is indicative of sometimes non-reabsorption of substances, and the substance then spills out in the urine. Appearance in the urine, sometimes a indicator or a sign of some disease or pathological process. In any case, there is a transport mechanism for different substances, a transport maximum, in fact. There are carrier molecules in the renal tubules that can be there, can act to help actively reabsorb different solutes in the filtrate. And if the carriers are saturated, then they are working at the maximum level to reabsorb whatever that particular target substance is, and they can't. They're saturated because there's so much of it in that filtrate that now the excess is excreted and starts to appear in the urine. Substances that are not reabsorbed normally are the ones that lack a carrier, or they're not lipid soluble, so they can't just pass. Lipid soluble molecules would pass unimpeded right through the tubule back into the capillaries, the peritubular capillaries. Sometimes substance is not reabsorbed because it's too large to pass through pores that exist in the tubular walls. Urea, which is the main nitrogen-containing excreted molecule, the main waste molecule that allows us to get rid of excess nitrogen, can in fact be partially reabsorbed. If the concentration of urea is high in the fluid that works its way into the collecting ducts, then some of it will get reabsorbed, but it then gets secreted back into the loop of Henle and eventually will go out. An important molecule to understand and be aware of in terms of molecules that are not reabsorbed is creatinine. Creatinine is a metabolic product from creatine phosphate. This is creatinine. Creatine phosphate, I remind you, is a phosphorylated molecule found in muscle tissue, which can provide a ready source of energy by the substrate level phosphorylation or transfer of a phosphate group from creatine phosphate to ADP to form ATP. And remember, I remind you that skeletal muscle fibers will in fact have a storage level of creatine phosphate, but once the muscle gets active, it that's only good for providing about 15 seconds at the most of energy to the muscle tissue. In any case, what happens is that creatine will be converted, and I'm not sure about the biochemistry of where and how, but it does come out of the cell and is either converted in the cell and comes out, or comes out of the skeletal muscle tissue and gets converted to creatinine. And creatinine in the plasma, in the blood plasma, will be filtered out of the plasma into the filtrate, and it is not reabsorbed at all. So with normal activity, there should always be routinely, regularly, a certain level of creatinine appearing in the urine. It gets filtered into the tubular system by filtration of the glomerulus, and it stays in there. It doesn't get reabsorbed. So in that sense, it's an excellent marker for the glomerular filtration rate, because if you measure creatinine levels, you can determine whether it's in the normal range. If it's too low, then that's an indicator for a inadequately, if it's below the normal range, then that's a very good indicator for a lower than normal glomerular filtration rate. So it's used as a marker substance and is monitored carefully and is monitored frequently in the blood plasma, that it stays within the normal range and doesn't start to accumulate in the plasma. Accumulation of creatinine would indicate that kidney function is inadequate. The other thing that does not get reabsorbed is uric acid. We do excrete some nitrogen waste in the form of uric acid, which is not a very particularly water-soluble molecule. We'll secrete very tiny particles or crystals of uric acid, and they appear in the urine, and they can be seen as crystals when urine is centrifuged down and you look at the precipitate, the centrifuged precipitate. Okay, now let's go on. I'm just going to pause for a minute. Okay, we're going to continue now with talking about regulation of concentration of urine and volume of urine. You should understand, and I've mentioned this before, I'm quite sure, that typically solute concentration in the urine is expressed in terms of osmolality rather than osmolarity. Osmolality measures the solute, is a measure of the solute particles dissolved per kilo of water as opposed to per liter of water. It also is a good reflection of the ability of a solution to cause osmosis. If you have a semi-permeable membrane, and all cell membranes are semi-permeable, then fluid on one side of the membrane versus fluid on the other side will move, and solutes might move, but some can move and some cannot move, depending on whether there are channels, and if there aren't, then instead of the solute moving, the water would move by osmosis. Now, as I said, osmolality is the measure because osmolality is per weight of water as opposed to per volume. Fluids change their volumes with temperature, so per weight, osmolality is a better measure. The kidneys maintain the solute load in body fluids at a constant 300 milliosmoles per kilo. And that's accomplished by the countercurrent mechanism that's present in the kidney tissue and through the help of the loop of Henle. Solute concentration in the loop of Henle can range anywhere from 300 milliosmoles to 1200. So at the bottom of the loop, it can be quite concentrated, up to 1200 milliosmoles per kilo. And the dissipation of the osmotic gradient is in the same osmotic gradient is also found in the surrounding medullary tissue, and that's prevented because the blood in the vasorecta will equilibrate with the interstitial fluid. So it's not as if it's not. So the vasorecta is an important structure also in maintaining that. Filtrate that flows from the proximal convoluted tubule into the loop of Henle interacts with blood flowing through the vasorecta, and you have a countercurrent type of exchange occurring. Here you see not only is the range in terms of the loop of Henle, the tubular system, going from 300 to 1200, but when you look in the actual parenchyma, the main tissue of the body of the kidney, you also see that gradient in osmolalities. So the loop of Henle, in fact, is acting as what's called a countercurrent multiplier. The descending loop is, as I've told you before, relatively impermeable to sodium, but permeable to water, and therefore water can move out into the tissue, and the osmolality will rise as the fluid moves down the loop, and then as the fluid moves up into the ascending loop and rises back up towards the cortex, the walls, the membranes are more permeable to sodium but impermeable to water, and therefore the sodium in there will move out but not water. Meanwhile, urea is still in there and isn't moving, and so urea gets concentrated in that way, and that's what we're mainly trying to get rid of. In terms of the collecting ducts, which are where the distal convoluted tubule empty into, and many nephrons, the distal convoluted tubules of many nephrons will empty into a single collecting duct, although there are many collecting ducts. But as the collecting duct moves down, there is some permeability to urea. If urea is very high in the collecting duct fluid, then it can move back into the tissue, but then it would get secreted into a loop of Henle, so eventually the body gets rid of it. Kidneys will get rid of it. Vasorecta is a countercurrent exchanger in that it helps maintain that osmotic gradient in the interstitial fluid, delivers blood to the cells in the area. It does a few things, but mostly it helps maintain that gradient. Here you see the osmolalities present in the tubular system. Here you see at the level of the glomerulus and the initial fluid going into the tubular system. Remember, it is the same essentially as plasma except without the protein. And then it goes through the proximal convoluted tubule and undergoes tremendous reabsorption. Most of it actually gets reabsorbed back. And then it arrives towards the loop of Henle as 300 milliosmoles, and as it goes down deeper into the medulla, the fact that the descending arm of the loop of Henle is permeable to water but not sodium causes the osmolality to rise up to as high as 1200, and then the ascending loop is permeable to sodium but not water, and therefore sodium can move out either passively or actively, moving out depending on where you are, but I'm not going to get into that detail. But sodium will move out and water stays, and therefore the osmolality will be reduced as it rises up, and it can even go lower than the normal tissue osmolality of 300. It can go as low as 100 when it gets into the distal convoluted tubule, and then it moves into the collecting duct. And in this case, this is quite a dilute urine going out down the collecting duct, down to the papilla and into the calyx and pelvis and out the ureter. So that's this. In this case, in this example shown here, there's quite a dilute urine flowing out. So this would be a high volume dilute situation where the person is putting out more volume of urine. The normal range of urine can be as wide as anywhere from 50 to 1200 milliosmoles per kilo, depending upon what the body needs at the time and what needs to be excreted. These values here show what's going on in terms of the vasa recta, the peritubular capillaries that are wrapped around the loop of Henle, and showing that the way in which the gradient is maintained, because in the blood, in fact, the osmolality is also, this range of osmolality is also maintained. Normally, osmolality in the bloodstream is always maintained at 300 milliosmoles, but here, these peritubular capillaries, they also have that wide range of osmolality, so that the gradient in the tissues in between, all this tissue in between, is maintained. Okay. I want to remind you that this is overlying this. They've just separated it in the diagram for ease of clarity, so it's not too crowded a diagram. Okay. So I was mentioning that was quite a dilute urine that was forming. Filtrate is in fact diluted in the ascending loop of Henle because of the pulling out of the sodium, and it's a very dilute urine in the end is created by simply passively allowing the filtrate to go out the collecting ducts and down into the renal pelvis, and then down and out to down the ureters to the bladder. Now that can happen. I think I might have talked to you about this before in 8A, but that happens as long as there is no antidiuretic hormone, or ADH, being secreted. I just want to remind you that ADH is secreted from the posterior lobe of the pituitary. I don't know if you knew that or not, but it's a hormone that gets not produced in the pituitary, but stored and can be secreted by the posterior lobe of the pituitary. But if the person is, if the person's physiological requirement is to put out a dilute urine, and that would occur if they had a overabundance of water on board, or their blood pressure was too high, then they would put out an increased amount of dilute urine. That can happen as long as antidiuretic hormone is not being secreted in the bloodstream. If that is not being secreted, then the collecting ducts remain impermeable to water. So no further water absorption occurs in those collecting ducts when ADH is absent, and the urine osmolality can end up being as low as 50 milliosmoles per kilo. It's all normal, from 50 to 1200, anything in between, all normal. It's what your body has to get rid of, okay? That's what it's doing. It's trying to get rid of it. So it's all perfectly normal, and it shifts quite rapidly. You could have quite a concentrated yellow dark urine, and then you drink a very large glass of fluid or water, a bottle of water, and boom, it'll change. The next time you pee, even only half an hour later, it'd be quite dilute. So the body's constantly shifting fluid and levels and adjusting to what's required to maintain a normal homeostatic balance. This is formation of dilute urine. In the case of formation of concentrated urine, what happens is that ADH gets released. Okay, so antidiuretic hormone would be released if the plasma osmolality is rising. Chemoreceptors detect a higher osmolality, then you get triggering of ADH release, antidiuretic hormone. That triggers expression, in other words, synthesis of and opening up of a protein called aquaporin in the walls of the collecting duct. It can be quite a fast response there. And aquaporins allow for water to move out of the collecting duct into the tissues where it can be reabsorbed into the peritubular capillaries, the vasa recta. In the presence of ADH, up to 99% of the water in the filtrate is reabsorbed. You can form quite a concentrated urine. Here you have formation of concentrated urine on the right side, and here formation of a dilute urine. We looked at this diagram before, increasing osmolarity as you go down the loop, decreasing as you go up, and then with no ADH it goes out as a concentrated fluid, as a very dilute fluid, low osmolality. But with ADH, antidiuretic hormone, diuresis, of course, is the loss of water in the urine, loss of increasing fluid. Antidiuretic hormone prevents that, and it prevents it by triggering expression of aquaporins in the wall of the collecting duct, reabsorption of water. End result, very concentrated urine going out. And you know from your own experience that you can have shifts between concentrated and dilute urine all the time through the day, depending upon what your activity level is, how much food you're eating or water fluids you're drinking. So, in general, diuretics, that's the term for substances that will trigger an increase in output of volume of urine, is any substance actually that's not reabsorbed. If a substance is not reabsorbed and it's increasing in concentration in the fluid in the tubular system, it can trigger it has its own osmotic potential, and therefore helps to draw water into the tubular system. Now, anything that exceeds transport maximum, for example, the transport exceeds the ability of the tubules to reabsorb, can act to pull water in and help hold water into the tubular system. So in general, these kind of things, things that inhibit sodium reabsorption will tend to act as diuretics. And diuretics can include, if a person has uncontrolled diabetes and a very high plasma glucose level, then the glucose will start appearing in the filtrate, and it could exceed the ability to reabsorb from the filtrate, and start spilling out in the urine, and water will be carried with it because of the osmotic potential of that glucose to hold the water in there in the space. Alcohol can act as a diuretic because it is a direct inhibitor of antidiuretic hormone activity. If it inhibits release of antidiuretic hormone, I'm sorry, I said activity, but it inhibits release of antidiuretic hormone. So if you don't have antidiuretic hormone, you're going to put out a dilute urine, a low osmolality, higher volume urine. It's a diuretic. And people know that. If they drink alcohol, they tend to pee a dilute urine. There are many other diuretics in our diet. For example, caffeine can act as a diuretic. It acts through a different mechanism. And then there are drugs. I don't know if these are still typically used or if they have second, third, fourth generation more modern drugs, but these are also diuretics, and diuretics are often prescribed for people who have a high blood pressure to increase the amount of fluid that they put out, so the volume of the intravascular space is lower, and therefore their pressure is lowered. Okay, We only have a few more slides, so I'm going to keep 6 more. So I'm going to keep going on this part 2. It's a bit long, but we'll finish it off soon enough. So renal clearance. I already talked to you about renal clearance, that it can be measured looking at creatinine levels. So when they talk about renal clearance, they're talking about the volume of plasma that is cleared of a particular substance in a given time. And renal clearance tests are in fact used to determine the glomerular filtration rate. It's a good indicator looking when they're looking to see if someone has glomerular damage, and it's also used to see if to watch or track the progress of a disease process in the kidneys that's previously diagnosed. Okay. So renal clearance is in fact equal to the concentration of a substance in the urine times the flow rate of urine formation divided by the concentration in the plasma. So the concentration times the flow rate, that's going to end up milligrams per mil versus times milliliters per minute. So you're going to end up with milligrams per minute divided by the milligrams per minute divided by the concentration of the substance in the plasma. Milligrams per minute in the urine divided by the concentration in the plasma. That is a measure of renal clearance. What else can I tell you about urine? It's anything from clear to pale to deep yellow, depending on the urobilinogen level in it. And that's a fairly good indicator of whether it's concentrated or not. Very concentrated will have a very deep yellow color. Some substances that are taken like drugs or supplement vitamins, some things in the diet can change the color of urine. If a person has a cloudy urine, that's one indicator of a potential possible infection. Although I can tell you that quite often if it's cloudy, it can also just mean that it's increased amounts of phosphate in the urine. It doesn't necessarily mean urinary tract infection. pH, typically slightly acidic. Range is 4.5 to 8. Kidneys are a very important physiological buffering system that allow for control, regulation, maintenance of a homeostatic balance of the body's pH. Specific gravity can range over this kind of a range and is all dependent on the solute concentration. Obviously, if you know what specific gravity it is, the density of a fluid, urine in this case, divided by the density of water at STP. Density of water at STP is 1 gram per mil. So when solutes are dissolved in the urine, the more solutes that are dissolved in the urine, the higher the specific gravity will go. It doesn't have units because it's density of the fluid divided by density of water. Overall general composition, 95 of urine, 95% water, 5% solutes on the average. This can of course shift the percentage of solutes. The nitrogenous waste exist as urea and a little bit of uric acid and creatinine, but also there is nitrates. There are some nitrates in it, but the main thing is urea. Other things that can be dissolved in the urine, normal solutes, include all of these charged molecules or electrolytes, and abnormally high concentrations of anything in there may indicate some sort of pathology, disease process in the kidney. It's a nice, easily accessed, easily collected fluid that can be monitored and examined by many different tests in order to and is therefore very informative about the health status of an individual. Okay, so now we get down to the bladder. The bladder is a smooth muscle sac, which has the ability to passively expand as it accumulates more fluid up to a certain maximum, but it reflexively, in other words, as a reflex, it reflexively relaxes as it fills, and only stops when it reaches the maximum, at which point the pressure builds, and that's when you get the urge to pee. Peeing is not particularly scientific, but you know, it works. People understand it. The more technical term is void, or dare I say, and you'll probably never hear this term again, I bet, micturate also means to pee. So as the urine accumulates, the bladder expands, relaxes, and expands. No significant increase in internal pressure. Males have around the bottom, underneath where the ureter comes out, they have a prostate gland wrapped around the ureter, which can cause problems if they get hypertrophy of the prostate. That occurs quite commonly in males as they age. Females, the bladder is anterior to the uterus. There is a region that is called the trigone. It's the triangular in the bladder. It's the triangular area outlined by at three corners by the openings of the ureters into the bladder and the exit point of the urethra. So you have this imaginary triangular area between those 3 points. It's important because infections tend to persist in that area of the bladder. Urinary tract infections, particularly bladder infections, often are quite common. And the bacteria most commonly associated with urinary tract infections, or UTIs, is E. coli. And the source is typically a fecal source. So that accounts for about 80% of all urinary tract infections. And from the lab we did, you're aware that E. coli, being a gram-negative organism, will have nitrate reductase and therefore be able to form nitrite, and that can be detected with the urine dipstick test. Little kiddies, infants, have small bladders. Kidneys are not good at concentrating the urine, and therefore they urinate a lot. They just let her loose all the time, constantly drinking water, constantly putting it out. The control of a sphincter around the urethra, and the sphincter in the urethra will develop as the nervous system develops. So it takes some time before little kiddies develop the ability to hold in their urine, which is why they need diapers. STDs, sexually transmitted diseases, can cause an inflammatory reaction in the urinary tract, and these are all general things, just sort of added points. One slide with some extra stuff on it. Kidney function declines with age. Many elderly actually become incontinent. They have difficulty being able to hold in the urine, and they'll spill urine.